When we talk about viruses, what happens? Do you feel fear? Do you think of diseases? Do you feel uncomfortable? But what if one of the most feared microbes on the planet was actually what could save us from the deadliest of illnesses?
What?
One of the most difficult challenges modern life science researchers are facing is the evolution of antibiotic-resistant bacteria. Through the process of natural selection, antibiotics actually act as a selector for bacteria that are resistant to antibiotics, resulting in the survival and reproduction of bacteria resistant to multiple types of antibiotics.
Whoa, now that's scary.
Take MRSA for example. What was once a susceptible bacteria has now become resistant to numerous strains of antibiotics, including penicillin, oxacillin, methicillin, and amoxicillin. This is where phages come in. Bacteria, being the most primitive form of life, has been in a constant struggle against viruses, both evolving and developing more and more complex systems in order to outcompete each other. Bacteriophages are viruses that infect and replicate within target bacteria. Its typical structure includes the capsid head, nucleic acid, collar, sheath, baseplate, spikes, and tail fibers. Phages generally follow two different cycles, the lysogenic cycle and the lytic cycle. In the lysogenic cycle, bacteriophages attach to specific receptors on the surface of bacteria and will actually combine with the host genetic information, but will not perform any actions because it is being repressed by special repressor genes on the virus. In about 10,000 replications that occur, the dormant phages repressor genes will weaken and become active, thus proceeding to the lytic cycle. In the lytic cycle, bacteriophages will first attach to cell surface receptors and inject their DNA with the help of ATP into the bacteria, thus allowing its replication. After entering the bacteria, phages will synthesize virus-encoded endonucleases that degrade bacterial chromosomes and instead replicates, transcribes, and translates the virus's components, which spontaneously assemble to form more phages. Finally, phages will release enzymes called endolysin that break down peptidoglycan in the cell wall, causing the bacteria to lyse, releasing the newly formed phages to be liberated into the surrounding environment to infect more bacteria.
Breakthrough time.
Phages are incredibly important, especially in phage therapy. In phage therapy, phages are collected, replicated, and stored to be used in treating bacterial strains. In contrast to antibiotics, which will kill most types of bacterial strains, both good or bad, phages will target specifically pathogenic bacteria without harming human cells or beneficial bacterial colonies. Phage therapy is so interesting because it has so many potential future applications, and it's definitely an area of life sciences that will continue to grow.
Thanks for listening. My name is Chen, and let's hope that this video goes viral.